Bismillahirrahmanirrahim. Assalamu alaikum. Welcome to my YouTube channel, Science for Everyone. Today the topic is related to virology, and the title of my today's lecture is that how virus affect the host cell. It is a very simple topic, but many students face problems in it. That, for example, if they are asked that how virus affect the cell, and what are the different ways that virus affect the cell, either it kills the cell or what does it do with that cell. So I'm sure that they will face different confusion in it. So just because of that confusion, I'm making this video just to make you understand and just to make a concept for you people that if a virus enters a cell, okay, it's suitable host. So which type of things, which type of associations, and which type of relationship will it form with their host cell, okay? Either it will kill it or it will make a peaceful association with it, or what else will it do with their host cell, okay? So let's start with the video lecture. Okay, this is a simple animated picture of it that when the virus enters the cell and when it is penetrated and the nucleic material is introduced into the cytoplasm. So after that, what are the different association that the virus is making with the host cell, okay? So if I zoom it a little bit, okay. So the first step is the adsorption to the host, and this adsorption and the entrance of the virus, I have explained it in the previous video, that how a virus enters the cell. If it is enveloped virus, so how it will enter. If it is naked virus, so how it will enter the host cell. But now the virus has found a suitable host for itself. For example, it is a flu virus. So what will it do? This flu virus will enters the epithelial cells of the upper respiratory tract, okay? Because those cells are the appropriate host cell of that virus. So for the first step is that the adsorption will occur, okay? After that, it will penetrate and it will release its nuclear material into the cytoplasm of the host cell, okay? So up till here, I have discussed it in the previous video. Now, the main topic of my today's video is that which type of association will it make with that host cell, okay? So the first is that we will just start from this acute infection, okay? We started from this acute infection. So just look here. Okay. First, if the virus enters the cell, so it will release its nuclear material into the cell, and those nuclear material will be replicated, and then it will be transcribed and translated, and proteins will produce, and many nuclear material will produce, and after that it will assemble, and new virus will be formed, okay? So if when it is formed, so this is rapid multiplication, okay? This is rapid multiplication of the virus. Just look here and the how much, one, two, three, for many progeny of viruses are made. So the first is that it will cause cell death and virus release. What will it do when it is concentrated inside the host cell? So it will then release, it will release from the host cell, okay? So what will it do by releasing from the host cell? So it will rupture the cell membrane of the host cell, and the cell will die and the virus will be released into the environment. And this case is very much related to the naked viruses, okay? Keep this concept in your mind. In case of the enveloped viruses, there is possibility that it will not kill its host cell, but it will just move away, just cross the cell membrane, taking a suitable cell membrane with itself, because those are the enveloped viruses, and the host cell will not that much be disturbed. But in case of the naked viruses, when it is releasing from the host cell, so they consist of different proteins that cause destruction in the host cell membrane, and after that they create pores in it and they are released. So when they create numerous pores in the host cell, so after that it is released and the host cell dies. Okay, so these are acute infection. This is known as acute infection. So the first is that it can kill the host cell, and it is known as acute infection. The second one is very important is, and it is very much dangerous. Okay, and this is the latent infection. For example, virus enter a cell, and after that virus is present but does not harm the cell, and this is very much related in case of the HIV viruses. When they enter the cell, so what does it do? They create a peaceful relationship with the host cell membrane, sorry, with the host cell. Okay, they lies in a dormant state in the host cell, although they increase their population, but uh, they are not killing that cell. They just remain inside that host cell. But keep this thing in mind, that the population with respect to the acute infection and the latent infection, the population of viruses varies. Okay, here the population will be more. If I write M, so it will be more, and here the population will be less in the latent infection. So keep this thing in mind, that in the latent infection, the virus is increasing its population, but in a very slow way. Okay, and it is not harming the host cell, and it lies in that host cell. And studies suggested that if it is not increasing its number, the single progeny of that virus is lie in that host cell. And the most important thing is that it can be activated. Okay, for example, if they get a suitable environment, and if they get an appropriate environment and appropriate signal. Okay, so what does it will do? It will transform into, it will be activated, and it will start its rapid production, and after that they will increase its population. And what does it will do? It will then kill the cell and it will be released. Okay, so this is latent infection. In case of the HIV, the HIV virus enter the host cell and it lies in that host cell. Okay, so in the early stages of the HIV, we cannot detect the virus, and it is very much dangerous because the virus is inside our cell, but it is not showing in the it cannot be monitored in the diagnostic procedures. Okay, so this is the second type of association the virus is making with the host cell. But keep this thing in mind, that it can be activated by different chemical factors. Okay, by different mutagens. Even studies suggested that it can be activated by sunlight. Okay, if there is a very high intensity of sunlight that is falling on us, so it can be activated. Different other chemicals are also involved in the activation of these viruses. Different proteins are involved, but I'm not showing you that, because then it will be a bit complicated. But just keep this thing in mind, that it can be activated and it can then increase its population. Okay, the third one is also very much important, and it is known as chronic infection. Okay, slow release of virus without cell death. Okay, the virus is increasing its number, but they are not killing the cell. They are releasing from the host cell, but they are not killing the cell. Here in acute infection, they are releasing from the host cell in a very high amount. So what will happen, that just because of it, they are killing the host cell by disrupting their cell membrane. But here the virus is releasing one by one. Okay, just look here. Here are 1, 2, 3, 4, 5, and many viruses are releasing, and here just only 1 or 2 are releasing. So this is known as chronic infection, because they are increasing their population, but they are not creating harm to the cell. okay, they are not destroying the cell. So this is chronic infection, although it is dangerous because the virus is increasing its population, and the increase of the population of the virus is very much dangerous, but it is not that much dangerous with respect to the acute infection, okay. So keep this thing in mind. The third is the chronic infection. The virus enter the cell, it increase its progeny, but they are not released in a very high amount. One by one they are releasing, okay. So it is known as chronic infection, and it is dangerous. The last one is, if I write, this is one, this is two, this is three, and the fourth one is the transformation into malignant cell, and this is very much dangerous because it is basically the virus is transforming the cell into a malignant cell, into a cancerous cell. So if someone asks you that can viruses cause cancer, so your answer will be a big yes, okay, because how they are causing cancer, I will make a video over it. It is a very that will be a very informative videos for you people, but here I'm just telling you that virus, there are different viruses that can cause cancer, and they can transform the host cell to a malignant, a cancerous cell, okay. So for example, a virus enters a host cell, okay. So those viruses consist of special type of genes that are known as oncogenes, okay. And oncogenes are known as cancer causing genes. So that genes are incorporated into the DNA of the host cell, okay. And after that, when it is transcribed and translated, so that proteins that are produced are cancerous proteins, and it can disturb the viral the host cell reproduction, okay. So basically when it is disrupted or it is disturbed, so the host cell can then divide, it can divide vigorously, and although we will the division is not required, but just because of the introduction of that genes, what will happen, it will transform into a malignant cell. For example, there is a virus and it consists of an oncogene, okay, cancer causing gene. So when the genome of the virus enters the cytoplasm of the host cell, so what will do, our ribosome will transcribe our host our RNA polymerase will transcribe that oncogene. After that, it will be translated, and the proteins that are produced are basically the cancerous protein. So that proteins can then interact with our normal proteins that are involved in our cell reproduction, okay. For example, there is a protein and it is used in the surveillance of the host cell reproduction. For example, that protein is responsible that at this time the host cell will divide, and at this time it will not divide. Or for example, if it is divided, so 4 cells will be produced or 8 cell will be produced, okay. So it is used as a, it is used to monitor the cell growth and the cell division. So what will they those proteins do, it will they will bind to that surveillance protein, and it will block its activity, so that protein cannot perform the surveillance. And after that, what will it do, when the host cell start dividing, so it could not be controlled, because the main protein that is used in the controlling of that cell growth and the cell division cannot function well, and after that the cell will start dividing unconditionally, okay. And basically this is known as the transformation into a malignant cell, okay. So the growth factors that are involved in the division of the host cell are disturbed by the proteins that are produced by the oncogenes, okay. Another type of gene is the proto-oncogenes that are basically present in the genome of the host, okay. So what will it do, the viral protein the wire for example, just look here, this is a gene, okay, and it is basically involved, this is a proto-oncogene, means that this proto-oncogenes is basically present inside the host genome. Okay, so when and this gene produce such a certain type of proteins that are involved in the surveillance of the cell growth. So what will it do? That the genome of the virus will go and will enter and will mutate this proto-oncogene. For example, if I change the color so you then you will be understand a little. For example, this is the gene the gene of the or the genome of the virus. So what will it do? It will incorporate itself in between this gene. Okay, so here just look here, this was a normal gene, and after that it replaces or it misplaces or it just incorporates itself into that normal gene that was producing the proteins that were involved in the surveillance of the normal cell growth. Okay, so after that when this viral okay, for example, this is the viral genome. Okay, and this red color was the this red color was the proto-oncogene. Okay, proto-oncogene. Okay, POG. So what will happen? This viral genome will incorporate into the proto-oncogene, and after that, when so the proteins now will cannot be produced. Okay, and if these proteins are not produced, so there will be no protein to surveillance the surveillance the growth of the cell and the division of the cell, and it will cause cancer. Okay, so this was all about that how virus enters a cell, and after that how in what in which ways does it affect the host cell. So it was the types of infections and their effect on the host cell. So I think hope you have understand this video. If you have any question related to this video, so just ask me in the comment box. I will answer you there. And if you have understand this video, so like this video, share it with your friends, and also subscribe to my channel for more interesting videos related to science. Thank you very much. God bless you.